Okay, so one of your classmates asked me, is the PowerPoint different? And the answer is yes. So I'm thinking I need to explain that. So we're on Module 2, right? Objectives and PowerPoints. So you'll notice that these chapters that we're going to go over are huge. And yeah, we're going to go over every last detail in every single one of them. So yeah. So I broke the lecture, instead of having this big, huge lecture, especially since I've been didacting them too, I'm going to break it into parts. So notice last time we discussed inflammation and we started on phagocytosis. We got about halfway through. So we're going to pick up at the end of that one. And then we're going to start the new one that I posted Monday night, which is the cells. Okay. And so then there'll be another one, right? So you'll notice that this one has 1A and this one is 1B. The next one will be 1C, right? And then, of course, the S stands for the student version, and this is the complete version that I use to present to you guys. It has everything filled in. So after today, I'll post the complete of this one. So the other thing I wanted to bring to your guys' attention is the test is over, right? I highly suggest reviewing it as soon as possible, and you need the lockdown browser to do that. So remember, if you don't want to install it onto your computer, there are computer labs on campus that you can use. The one we used for testing, if there isn't a class in there, you're more than welcome to go in there and utilize that lab, the Hibernia Center, Building 1. Or upstairs here on the second floor in the Big Fish Bowl, right, the open lab. You do have to have your student ID, though, to get in there. They swipe it. As opposed to the Hibernia Center where you just sign in with your access code. Any questions on that? Okay. So again, we have to have everything closed on your computer, open up lockdown browser, it'll take you right to Canvas, you log in, go to the test, and you should be able to see your results. So it will actually let me do that without being in lockdown browser. So, test 1. Teachers can override these kinds of things, you know. But you should, it should be right below your test, right? I think, clicked a few things, so I mostly got them wrong. But what I wanted to show you guys is that you'll see the question, and if you got it wrong, right, it'll show you what you answered and what the correct answer is. But also what I would really want you guys to look at is the objectives, right? Because I'll change up these questions and such. I'm not going to ask it the exact same way on the final exam, right? Any of these objectives that made it on to the test, you have a 50-50 chance of seeing that objective on the final. So again, I'm not giving you a final exam study guide. You already have it as your test, right? Your tests will always be available like this once everyone's taken the test. Okay? Make sense? All right, so look at and create your own study guide, right, based on how you did on your test. I guess I think I purposely answered a couple wrong so that you could see what it looks like when you get it wrong. Right. I should hopefully get a perfect score on my own test. All right, moving on. Let's pick up where I want to go to my modules because I am going to access probably animations today. I can't remember. No, we watched the videos last time. We did do that. But just as a reminder, right, these animation and video links, right, so we've watched all of these. I do remember that now. Whether we watched the actual phagocytosis one, I can't remember. We might watch it to review. There's actually a new cool one that I like that's with a new book I'm looking at. So, innate phagocytosis. So let's just watch it to review. What the heck, right? Wake us up a bit, and we'll fill in the details. I'm thinking audio might be good. We'll soon find out in a second. so I thought it was good. You ever like touch your touch screen of your phone and it just doesn't react? The touch screen of this panel is not reacting to me increasing the volume. Please don't say that's going to be one of those days. So let's see if that will help us. I just doesn't want to go up. I'm not restarting that system.
In phagocytosis, phagocytes are attracted to the area underneath the chemical products of the microorganism. Phosphates released by injured mammalian cells, or like components of a complex. The phagocyte moves into the area of adhesion and then attaches to the microorganism. The attachment is mediated by a variety of surface receptors, including antibodies, lipopolysaccharide, and complement receptors. For example, the C3B component of complement bacteria or other particles and then binds to C3B receptors on the phagocyte. This process of coding to enhance phagocytosis is called opsonization. The microorganism is then engulfed by the phagocyte into a vacuum known as a phagosome. Vesicles in the cytoplasm called lysosomes fuse with the phagosome, releasing digestive enzymes such as lysozyme and proteases into the phagosome. The structure resulting from this fusion is called a phagolysosome. Inside the phagolysosome, microorganisms are killed and digested. Finally, the digested contents of the phagolysosome are eliminated from the phagocyte by exocytosis.
Stay there, yeah, I'll stay there. Okay, so how many of you guys looked at the new PowerPoint that I posted Monday night and read up on cells? Woohoo, yay. One indication. Okay, so they're saying phagocyte, right? They're not being specific as to what white blood cell is doing this, right? In this video, they're being general, which is great. But those of you that may have taken anatomy and physiology 2, any of you guys in A&P 2? One person, okay. You looked at white blood cells under the microscope, right? So you should be able to identify them by their distinguishing features, which thankfully you don't have to do for this class, right? But you know, that knowledge can be handy. And so based on that knowledge, right, I see a single nucleus, a pretty large cell. Any guesses as to what you think it might be? It could be a monocyte, and then monocytes differentiate into more powerful phagocytic cells in the tissue, and they're called what? They're called macrophages. Oh, okay. Some of you guys know this stuff, right? Where if this was another main phagocyte, which can anyone think of another one where their function is phagocytosis, to engulf and digest stuff like we saw here, it would have a multi-lobe nucleus. You're right. Don't question yourself. Say it. Neutrophil. There you go. Neutrophils. Right? And so the clue here is that the nucleus isn't that multi-lobe shape that we characteristically see for a neutrophil. But they're major phagocytic cells. And that was the one, remember, we watched in the video where they just took blood from a graduate student, put it on a microscope slide, and the neutrophil went and chased down and ate the bacteria they put on that slide. Those guys, as we know, are very important because they're going to literally ingest and destroy pathogens like bacteria. So in order to do this awesome process of phagocytosis, which some of our cells are really good at, like the macrophage and the neutrophil, which are our two main phagocytic cells, they have to know where to go, right? Remember, we said that this is very specific. When we elicit inflammation, there are chemical mediators that are released that are going to let the cells know to go there, right, to that specific spot. So the very first step for phagocytosis to happen is the cells have got to know where they're going, right, and head to that particular area. And since they're responding to chemical signals, we refer to this as chemotaxis, chemo referring to chemicals, right, taxis, movement. I always just think of a taxi cab, right, and helps you remember that that's movement. So they're recruited by these chemoattractant, by these things that say, come here, this is good stuff. So the microbes themselves, as we heard in that video, and in the animations and stuff we watch, they release things that are going to track the phagocytes. The phagocytes can sense certain components of bacteria that are released by them, and it's going to track them to them. So phospholipids from our injured cells, right? As our cells get injured by a cut or a splinter or the microorganisms attacking our cells. Chemokines is a whole class, a whole bunch of chemical messages put out by the immune system that say, come here, right? Because chemo refers to, again, it's a chemical signal. Kines refer to cytokines, all these chemical messages that the immune system uses. And then in the animation and in this diagram, they have a particular molecule known as C5a. The C stands for complement. And complement, remember, is a protein, several proteins, excuse me, that are found in your blood. And they're going to leak into your tissues during inflammation. This particular protein, the C5a protein in the complement system, is a chemoattractant. It calls the phagocytic cells to that area. And unfortunately, that's one of the ones you really need to memorize. The C5a, yeah. Oh yeah, we'll get into that one. There's just a few, right, that are really key players in complement that you guys will need to memorize, and this is one of them. So, okay, so we're calling them there, right? But just because they're there doesn't mean that they know exactly what they need to grab onto. And that's where the being able to recognize and attach is important. And so again, another complement protein, as I discussed in the animation, can aid in this process. It is called C3b. And I always remember B for binding, because it binds to the surface of pathogens like bacteria. And because it binds to the surface of the bacteria, so this is the bacteria, this C3b is a protein we produced that's sticking to them. It's helping us identify, but it even goes one step further. It helps us be able to attach, because our phagocytic cells have receptors for C3b. When it's attached to a bacterial cell like they're showing here, our phagocytic cells literally grab on to the C3b. Because this substance binds to the surface of the bacteria and helps us recognize and attach to it, this process of recognizing an attachment is called opsonization. And our antibodies do this too, right? When our antibodies, which are also proteins as well, when they bind to a bacterial cell, our phagocytic cells have receptors for our antibodies. And we're going to be able to therefore recognize that this is a bad guy. We're going to be able to attach to it. And then, of course, the next step is it's going to induce that cell to do what? What is it doing here? It's going to engulf it, right? That's our next step, is engulfment. So the pseudopods, just like amoeba, engulf their food, right, and also move. They're going to push the cytoplasm, surround that pathogen in a membrane. That vesicle has a special name. It's referred to, in this case, as a phagosome. I was talking with somebody last night. People from Britain, right, even though it's the same language, it's a different vocabulary. They call a cigarette a fag. We were saying how much crap that exchange student took all the time for asking people for a fag. But, yeah, so phagocytosis, but in this case means to eat. I have no idea how a cigarette got called a fag in Britain, though. I don't know the background. Oh, because like a stick? Oh, fag means stick. I knew there was some connection there. I just couldn't remember it. So again, words mean other things than what we realize, right? So this is, where medical terminology and stuff like that really can help you out, and knowing roots of words, like the fact that fag also means stick, in this case it means to eat, okay? So they're going to engulf it in that vesicle, and then that vesicle is going to fuse with lysosomes. And lysosomes, remember, are vesicles that contain digestive enzymes. So those lysosomes are going to fuse with the phagosome. So guess what they do with the name? They fuse it, right? It becomes a phagolysosome, right? Put the two parts together. And then destruction is going to happen, right? So in those endosomes, right, when this fuses, the pH drops significantly, becomes very acidic, right? And those digestive enzymes just start destroying what it is that's inside that vacuole. And remember, this is a membrane-encased vacuole. And so we're keeping it contained, right? We wouldn't want this happening within the cytoplasm, right? Because then we would digest our own proteins and our own components. So we do this very securely inside that vesicle. So we're going to destroy it. We're going to use toxic substances like noric oxide. As I said, the pH is going to decrease significantly. It's going to become very acidic. The enzymes are going to degrade. Defensins are going to damage the membrane of the invader. So this is a substance that we produce. They're proteins or peptides. They're not quite long enough to be considered proteins, but made of amino acids. And they literally stick into the membranes of foreign cells and disrupt them. Lactoferrin, again, it has ferrin in its name, refers to the element of iron. And so we're going to really tie up iron because, again, iron is very important for metabolism, for pathogens, for microorganisms, for bacteria. And so we want to sequester that iron. And then the vesicle is going to fuse back with the membrane, and it's going to expel the remains. So this is a pretty awesome system, right? Why is it that we don't kill every bacteria that gets into our body? Why don't they get eaten up by phagocytes, by our macrophages and neutrophils? Why do we still have disease? Remember back for our macrophages, one of the things that we looked at, no, haven't looked at yet, never mind. Did we look at the tuberculosis granuloma? We did, okay, I thought we did. I thought we did. Let me go back to that slide. I was like, all right, I got to do the quick way. Yeah, there it is. I wasn't losing my mind. We have looked at this already. So what's happening here, y'all? What is this cell right here? It says it's a giant cell, but what is it? It's a macrophage. The little green things are the bacteria, in this case tuberculosis, Mycobacteria tuberculosis. That's a macrophage, right? These are macrophages. What are they supposed to be doing to that bacteria? They're supposed to be killing it. But are they in these pictures? No. They are not successful at destroying this mycobacteria. This is why we have disease, because even though we have all these systems that we're going to talk about and all these awesome cells, there are microorganisms out there that know how to beat the system, right? I've got a dog. I had a really awesome dog, but he died. But I got another one. She's cute. She's got a pretty ferocious bark, right? That's a pretty good deterrent for someone coming into my house, right? I've got an alarm system. I've got a lock that I actually use, right? These are all deterrents. Does that mean that there's no way I'm going to get robbed? No. If someone is pretty darn determined they're probably going to try and come in. I've got a gun too. I'll take care of that. Right? So I have all these levels of defense, but there is still the possibility, right? Someone's going to get in and take my stuff. Of course, I don't have stuff. That would be my husband who has stuff, right? But you see how we have all these defense mechanisms in our bodies, right? My old dog, if you hopped the fence, he'd eat you. I kid you not. Joy, I don't know, she might just bark at you. I haven't found out yet. I don't want to find out. But we've got these phagocytic cells, and their job is to eat, right? But there's going to be times where they're going to get tricked, right? And they can't figure out how to eat it, right? So tuberculosis is a good example here of, like, we can't eat it, right? Our cells don't know how to eat it, don't know how to destroy it. Like, we don't have the enzymes necessary to destroy this guy. That's why we have this disease, right? We can't get rid of it on our own. What do we have since it's a bacteria, though? Someone has tuberculosis, we just go, okay, just going to eventually die, oh well. We have antibiotics, right? We do have antibiotics against tuberculosis, right? But, this is why treatment's difficult and long-lived, right? Is they'll hide out inside of our cells, not being killed. There are other infections that you can have. Let me go the fast way and get back to my main picture. So, there are some, Shigella, for instance. It'll get inside, it'll purposely actually, it does things to purposely get engulfed by our phagocytic cells. But then once it's engulfed, it can get out of the phagosome. And then it can even use our proteins, our actin filaments inside of our cells to slingshot itself into the next cell. Evil bacteria, right? There are others that, the phagosome, like with tuberculosis, right? The lysosome fuses, they don't die. They keep growing, actually, inside that phagolysosome. None of the bombs that we throw at it are effective. None of the enzymes, nothing. It just laughs at us. But, the good news is for most of the invaders, we successfully eat them and destroy them. Right? But we still have disease, we still have infection, because some of them will get past this. Some of them actually even avoid getting engulfed to begin with. Right? So you see how these guys got coated? Remember an outermost covering that bacteria, some of them have, that we say helps protect them? Anyone remember what that layer is called? In lab, we stain the background in the cell so we can see that unstained extra layer that some bacteria have. It's made out of sugar, usually. No, peptidoglycan's a cell wall. Capsule. A capsule. So things like Klebsiella pneumoniae, huge capsule. That's why we use it in microbiology lab to stain it, so students can see that huge capsule. It sticks really well to our cells, and it evades phagocytosis because we don't have anything that sticks to that capsule. We don't see it. So the very first step, or second step that we need to happen, recognition and attachment, can't happen for encapsulated bacteria. So as you can imagine, a lot of the ones that make us really sick, guess what they have? A capsule. Helps them avoid this destruction. So as we kind of alluded to earlier, our two main phagocytic cells, where that's their number one job, are our macrophages and our neutrophils. So we're going to do a little compare and contrast. So macrophages are always found in our tissues, and they're derived, as we said earlier, from monocytes. So they're called monocytes as they're circulating through our blood, and then when they leave our blood to go into our tissues, they become more powerful, right? Their job changes a little bit. They differentiate. and they stay in our tissues. We have resident ones all throughout our body, and when they die off, they're replaced from the monocytes in our blood. Unlike neutrophils, granted I wrote only here, really just very rare are they in our tissue unless they were called there during inflammation. Right? They don't just go there and hang out like macrophages do. They're called to the scene. So can anyone tell me anything else about a macrophage or a neutrophil? Macrophages live longer. So therefore, by default, neutrophils don't live quite as long. I've got some nicknames for these guys. Anything else? Right, a macrophage slowly wanders or remains stationary. Who's the better killer? It depends. It depends. Right? So, these guys, the macrophages, will survive after phagocytosis. They keep regenerating their lysosomes. And that's why they live so much longer. Where degranulation, because their lysosomes are sometimes referred to as granules, neutrophils will die. After they've used up all their granules, all their lysosomes, they die. Macrophages can be activated by T cells and increase their killing power. Right? So they can get really powerful, but they need help. They need some confirmation signaling from T cells. Where neutrophils have the greatest killing power straight up, right away. But this comes at a cost, right? To be so powerful can be very deadly. Macrophages will fuse together to form giant cells, right? When they can't kill the invader, they're just going to keep fusing with one another to try and keep it contained within the cell. And then granulomas are when you have that much larger group structure of giant cells, maybe even necrotic phagocytic macrophages where the bacteria are leaking out, where you'll have neutrophils even coming into the equation, and you'll have T cells regulating what's going on. Right, so this ends up being a very large grouping of cells, and depending on what's going on, right, will determine how that granuloma grows and whether we're going to be effective with treatment. Neutrophils can help out too when we've got a serious invasion on our hands. They can shoot their DNA out of their cells. So they're going to kill themselves in this process. But in that case, all those fibers of the DNA create a net. And we can help trap the invaders in a particular area. So the next thing that we're going to look at, which is actually going to bring me to my next PowerPoint, is we're going to dive in more into the individual cells and their functions, their jobs. What do they do? How do they help us out? So this is a pretty good table that's in your book that summarizes, right? It's not complete. Right? It summarizes the major functions of the major groupings of cells. So one of the things I want to point out to you guys is that we, again, group cells. So there's a group of cells referred to as granulocytes, right? They all have something in common. And you have your mononuclear phagocytes and your lymphocytes, and sometimes neutrophils dendritic cells are given their own little group because they're pretty darn special guys. So I'm going to switch over to the newer PowerPoint that I posted for you guys, Part B. the cells, right? So before we dive into the cells, there's some terminology that I'm going to use, so I want to make sure that we have a working definition of it. So these are the definitions straight from your book, and then, you know, I'll add some helpful information to them. So this is really important. For innate, innate typically means what? It's already there, right? You're born with it, right? So as I have in parentheses there, this ability is encoded in your DNA. You have this actually even before you're born, right? Because your DNA determines your innate immunity. So your innate immunity, what you're born with, are standard host defenses, anatomical barriers, right? So we can keep the bad guys out. We have sensor systems, built-in sensor systems. So when the bad guy gets past the wall, right, past that barrier, a sensor's tripped and an alarm goes off, right? So we're able to recognize specific patterns associated with microbes, with the bad guys. Where unfortunately most house alarms, you could set that off yourself, right? It doesn't know you from the bad guy, it just knows someone crossed the threshold. And the alarm goes off. Although we don't want false alarms, right, in our system. So the sensors are pretty specific to bad guys. And then the phagocytic cells, right? These are the coppers on the scene. They're going to destroy the bad guys. They're judge, jury, and executioner all in one. They ain't messing around. And then, of course, the inflammatory response of getting cells to the area where invasion has happened. So they can destroy it. So they can try to contain it. This ability is encoded in our DNA. This is not something our body learns. This is something our body does day one. Unlike our adaptive says what? What does adaptive mean? Yeah. But very simply, adaptive means to be able to what? Adapt. What's a synonym, a thesaurus word for change, right? You can change, right? So although you're born with the basics, right, on basically how to make this system run, it's going to adapt. It's going to change over time, right? So it provides protection that improve due to exposure to antigens. And the major players, the major cells that are responsible for this are our B cells and our T cells. And these are actually lymphocytes, but we commonly abbreviate them by these single letter terminologies. So I just said another word, didn't I? Antigen. Who knows what an antigen is? A and B what? Oh yeah, right? So like on our red blood cells, right, we refer to those A and B antigens which determine our blood type. And these are antigens not for us, though. We would call that a self-antigen, right, if you have that blood type. But if you have AB blood and you stick it in me, it's going to be bad news because I'm O, right? I don't have those antigens. So my body's going to see it as foreign and attack. And that's the important thing about an antigen. It is very simply, it's something foreign to your body. Your body does not recognize it as self. And therefore it's going to attack. So even though these are human red blood cells, right, they could have stuff on them that my body has never seen before, and so it's going to attack. The word antigen actually means antibody generator. So in the definition, you'll say, the molecule that reacts specifically with either an antibody or an antigen receptor on a lymphocyte. So this is something that gets into our body, something foreign. And it can even be something on something, right? Like in the case of red blood cells. And our body can make antibodies against it. Or we may have receptors on, say, the T cells that will recognize it. We're going to physically interact chemical to chemical, molecule to molecule. So who knows what an antibody is? Yeah, it's a protein that binds to the antigen. Where do they come from? Nope, close. B cells. B cells make them, right? They're a product of our own immune system, right? They're protective proteins that we made. They have a characteristic Y shape to them. It's a protein, for the most part, it's protein, and it's going to bind to that antigen. And it was one of the first things that immunologists could study, right? These get pumped into your blood, right? Proteins are pretty soluble. They travel throughout your blood to help protect your body. And so when scientists looked at blood, they were like, oh, what is this stuff? And, oh, look, it's sticking to other stuff. Why is it doing that, right? Why is it binding? And they found if they put foreign substances in somebody, they produced antibodies against it. So hence, again, the name antigen, antibody generators, right? If you put something foreign, you get an antibody. And that's how we actually make antibodies. When we purposely want to make antibodies against something, we stick it into an animal, and it'll make antibodies against it. And antibodies themselves are proteins, right? So one of my favorite shows, though, although it kind of got knocked off the pedestal, because, you have to, when you cohabitate, you have to agree with the other person about what shows you watch. But my husband didn't really care for Scorpion, but I thought it was pretty cool. But they made me really upset one night last season because somebody got bit by a snake, right? And so they went to where there were snakes housed, all dramatic and everything, and get the venom from the snake. And then they put it into, can't remember what it was, probably a poor guinea pig. Rabbits are used a lot, though, for antibody research and stuff like that. So it put it into another animal, right? And got it to make antibodies against it to be able to give it to the patient, right? So antibodies against the venom so that the person's body, right, would recover from getting bit by this snake. The only problem is that this all happened in a half-an-hour show. It can be done, and it is done like that, right? So they actually put it into pigs. Pigs are more like us, believe it or not, than any other animals out there. So those antibodies that pigs make, we don't actually see them as pig antibodies. They're close enough to ours that we don't even respond to the antibodies being foreign, because we don't want that, right? We want our body to think it's our antibody and deal with it our way. Back in the day, they used to use horse serum. So the antibodies from the blood of a horse. Horses will actually get bit by snakes and survive. They, I guess maybe it's body size or a difference in immune system. They'll make antibodies. And so you can get them from horses, and they would give them to people to save them from snake venom. The problem with that is it takes about how long, I think we've discussed this, right? When you get the flu vaccine, right, they're basically giving you the virus, modified virus, to get your body to make what? Antibodies. How long does that take? A week or two. So 9.5 hour show. You see my problem with it? Ain't happening. They ain't saving that dude like that, right? What they were better off probably doing is breaking in to the section where they keep the anti-venom. Because anybody who works with venomous snakes, right, at the herpetology unit, the snake house, right, over at the zoo, trust me, they've got a case of anti-venom on hand in case somebody gets bit. Right? They could have just stole that. It's pretty cool that they tried to, bring into the science of how you make antibodies. It just can't be done in half an hour. It takes a while. Make sense? Right? So, it's done all the time. So we're going to actually go through that process of talking about how it is that the immune system, recognizes that antigen and makes antibodies when we get to the next chapter, which is adaptive. But these guys in the beginning, right, of recognizing and destroying the invaders are really going to help us, right, hopefully keep us alive during that week to 2 weeks that we might need to make antibodies to completely eliminate the invader. So our cells, these white blood cells that we're going to talk about, and of course always the red blood cells, are always found in our blood. But white blood cell numbers are going to increase during infection, right? Because we're just going to need more of them. The normal numbers that we keep on hand is not going to be enough, right? So the same thing here, right? In the United States, if someone invaded the United States, what are we going to do? We're going to call up the reserves, right? We've got military personnel on reserve, right? We've got National Guard that are meant to protect the nation. Are they full-time military personnel? No, they're part-time, right? But they're trained and at the ready in case we need them. So we got cells, right? Where are they hiding out in our body waiting for their call? They're in the bone marrow, right? They're in the bone marrow, right? And that process of making those cells is referred to as hematopoiesis. And there's specific yes, that word. There's stem cells, right? That are going to become any one of the blood cells that we need it to become. How does that cell know that we need neutrophils or that we need macrophages? How does it know that it needs to turn and have its descendants turn into that particular type of cell? Right, chemicals. Remember that this world of our body is all chemical, right? So there's specific chemicals that say we need this group of guys. So that group of chemicals are referred to as colony stimulating factors, right? We need that particular colony. We need that particular group called up, right? We need those specialists because we're dealing with this situation. And that really happens. Doctors are going to do sometimes where they're going to take your blood and they're going to run what's called the differential, where they're actually going to determine the percentage of all the different blood types, all the different white blood cell types in your blood. And if we have changes in the different populations, that kind of gives us a clue as to what's going on. So like if you have overall more white blood cells than you normally do, right there is a sure sign of infection, right? Why do you have all these white blood cells in your blood? This isn't normal level. This is above normal level. Okay, well, who are the major players? Who has increased? Is it neutrophils, right? And that's usually a pretty sure sign of bacterial infection, right? When you got a lot of neutrophils, right? Because you got to do a lot of killing, right? But, what if we got a whole lot of lymphocytes? Right? We've progressed to a different stage, right? Adaptive immunity is on the forefront now. It's really kicking into high gear. So, obviously in your bone marrow, you're going to make buku red blood cells, right? Very short lifespan for these guys. Long distance truckers, they get beat up along the way. But they really don't help us at all with immunity, right? Their job is to carry oxygen. And in the case of malaria, they're actually kind of our downfall too, right? Because the plasmodium infects red blood cells. But we kind of can't live without them, right? That whole oxygen-gas exchange thing, a little bit important for life. Right? We're aerobic. That oxygen thing, it's got to happen. Platelets, these guys really are going to help us out as far as infection goes, and especially with damage to blood vessels. They're like little sandbags, right? They're going to plug up the holes, right, when we have destruction to blood vessels. And they're actually not cells. And this is why in anatomy and physiology, right, they refer to these things as formed elements. They don't say the cells, because these are actually pieces of a much larger cell called a megakaryocyte. And so, like I said, kind of like little sandbags. Except instead of just having sand in them, they actually have enzymes in them that will help activate the clotting cascade. Right? So the proteins that are already in our blood too, that are going to help coagulate the fibrin, they're going to form that clot. Those are going to get activated when these little sandbags get busted open, when those enzymes get released. And although, right, they're going to help protect us by like walling off the area and stopping blood flow uncontrollably into our tissues, that's pretty much it as far as protection. So the good news for you guys is we're not going to go into the clotting cascade and all that stuff in this class. They can do it in anatomy and physiology. We're going to stick to the really cool guys, which are the white blood cells. And then the white blood cells, right, you can tell they're my favorite. They're pretty cool. And they're beautiful under the microscope, just beautiful. So I'll typically use the term white blood cell, and I won't say leukocyte, although that's another term for white blood cell, because that whole site ending, right, some people confuse with lymphocytes. And lymphocytes, I see both these words start with L, both of them end in site, but that beginning part really is important. When you say leukocyte, you're talking about all white blood cells. When you say lymphocyte, you're talking about a particular group of white blood cells, right? And a lot of times, you know, back in my days of teaching anatomy and physiology, you guys would switch up these two terms all the time, right? And even, remember, I commented on one of our videos, and she said lymphocyte, homie. I'm like, no, no, no. I don't think that's B cells and T cells heading to that area, right? You just literally injured that poor zebrafish. That's neutrophils and macrophages coming on scene, right? You probably meant to say leukocytes, but why not just say white blood cells, right? Less likely to screw it up. So within the white blood cells, we have four major categories that we address. What we collectively refer to as granulocytes, mononuclear phagocytes, as the name implies, right? They have a single nucleus, and they eat stuff, right? They're phagocytic. Dendritic cells, as I said, sometimes get their own designation, but also sometimes are lumped in with mononuclear phagocytes. And then you have your lymphocytes. So, what do we need to focus on these guys? We need to focus on what they do, what their functions are, right? But we still need to know how they're classified. So it all starts in the bone marrow, right? The bone marrow is going to get signaled as to who we need, right? And so then they're going to differentiate into the different groups and into the different lineages. And I don't know if you guys can see this up on the screen, but you see this box, right? So this is denoting mature cells, right? These are the ones that leave the bone marrow, right? Same thing here, right? This is a mature red blood cell, and then you have your platelets, or sometimes known as thrombocytes, which are pieces of this megakaryocyte. They're not actually cells. In mast cells, they kind of got them drawn like this, boom, boom, because they spend a very short period of time in the blood, and they travel right to the tissues where they hang out. And there's a distinction here too in the diagram that notice that, again, a monocyte is what we would call this cell type in your blood, right? If you took a blood sample, you looked at it under the microscope, you'd refer to as a monocyte. But when it travels into your tissue, it can differentiate into a macrophage, right? These really good killers, eaters, mega big eaters, right? Macro, meaning big. And then it could also maybe turn into a dendritic cell. So this is denoting in our tissues, right? Not in the blood, the second line. That's why there's this distinction. And the thing is, too, is that T cells, too, will differentiate in our tissues into effector T cells. The major differentiation that's going to happen with B cells is they're going to turn into plasma cells. Not to say you can't find these in your blood, right? It's just that that's not their main house, right? If they're in the blood, they're traveling. They're traveling. They're just hopping on the superhighway. So granulocytes, I even stole from the web pages that I first sent you to. These guys all have in common that they contain cytoplasmic granules. And really the important thing here is that they stain, right? When scientists do a blood smear and they stain these cells, they all have granules that stain for the most part, and we're able to visualize them under the microscope. So here they had fun, the artist had fun, and drew the granules as freckles, which I have freckles, so I found that. Common element, right? So yeah, I mean, it's creating a distinction for them. So we have 3 granulocytes. What are their names? Right. So what's this one, though? That one's a neutrophil. What's this one? Basophil. What's this one? Eosinophil. Why do they have these names? What do you know about their granules? They stain differently, and that's how they get their names. Also notice, let's put the names up. All their names end in what? Fill. Because they're all filled with granules. And the first part of your name is going to tell you about their granules. Neutrophils, a lot of times, depending on the stain and the age of the slide, you're not going to see their granules very well. They're relatively neutral in their staining, hence the name neutrophils. So notice how they're kind of light here. Basophils, on the other hand, are named after the fact that they're stained with the basic dye, and the most commonly used one in the dye mix that's used to stain these cells is methylene blue. So they'll have a bluish colorization to them. Eosinophils, on the other hand, are going to stain what color? Color do you see down here? Kind of pinkish, right? Sometimes it'll be kind of orange. It's more, eosin is a red dye, right? And eosin is the one that gives them this reddish to orange color. If it's a really good stain, it's usually a nice, good red color. But again, that's why they're called eosinophils, because they stained with the dye eosin. So what does that tell you about these granules, which are basically just, vacuoles like lysosomes? Especially in the case of a neutrophil, it's probably lysosomes, right? So digestive enzymes. So they can do phagocytosis. But basophils and eosinophils, they stain different colors. So are their granules probably what? Different. Right? They're a different color. They got stained by a different dye. It means there's different stuff in there. They do different things. It's that simple. So basophils, for instance, people like myself who experience allergy, we're not so friendly with these guys. Right? Basophils are bad news for us. That's my nickname for them. because they contain substances like histamine, right? So those granules, when they release those substances, one of them could be histamine, which is a vasodilator, makes the blood vessels permeable, and you get inflammation in that area, right, where they've released their granules. So this is why people who experience allergy, right, when you're exposed to something that's going to set off these cells to get them to release histamine, you take antihistamine beforehand, right? So it's already in your system. So if your friend has a cat and you're allergic, you take your allergy medicine before you go to their house, right? The antihistamine's in your system, so when the histamine gets released, guess what? Nothing happens. The antihistamine, right, outcompeted it, and you don't get that reaction. Some people are like, oh, my allergy medicine's not working, because you already had the reaction and now you're taking it. It's preventative medicine. Some of it has components, right, that are going to help you decongest afterwards, right? They're going to help you with the fact that you didn't take it beforehand. But you really are supposed to take it before. Well, if you're allergic to like everything outside like myself, you take it every morning before you leave the house. And those days you forget, you know it. Mast cells, right? Remember those guys we drew a line down when they're in the tissue? They contain histamine as well. So they're kind of close cousins to basophils, except as you can see by the lineage, if we go back to the lineage chart, they're not derived from basophils, right? They have their own lineage. But they're similar in chemical makeup, right? So when you stain them, they're going to come out similar. They both have histamine. But these guys are in your blood, and they'll travel into your tissues, where mast cells live in your tissues. So then eosinophils, we find these guys really increase with parasitic worm infections. And again, due to the fact that their granules are effective against fighting those types of invaders. You'll also see them increase in allergic reactions. But that's because these are guys that are the counterpart. These are the guys that once, the basophils release their histamine, right, and they call everything on scene, they cause that inflammation. Well, eventually that needs to stop, right? So the eosinophils are kind of the regulators. They'll actually destroy excess histamine with an enzyme called histaminase. And they have other antimicrobial substances, so they're helpful with certain types of infections, especially, like I said, the parasitic worms. Thankfully, in people, right, we pretty much know where parasitic worms come from, and we avoid infection nowadays. So you don't see that quite as often. But that's definitely like when a doctor does a differential and eosinophils are off the chart, they're like, whoo, we might have some worms on board.
Like, say if you got a parasitic worm, would you need to take your allergy medicine?
No, it doesn't work that way. So he said if I got a parasitic worm, would I not have to take allergy medicine anymore? No, because the eosinophils would be all about the worm, and they wouldn't be dealing with my allergy issue. So that would be a bad case scenario. My allergies probably be worse with a parasitic worm infection, because I wouldn't be quieting down the allergy attack because my eosinophils would be so busy with the worms. Our system isn't very good at multitasking either, right? Which is why, like, sometimes when you'll get one infection and then you get another infection because your system got kind of wiped out by the first infection and you didn't recover before something else invaded. It's just like in war, right? If you don't have enough troops, you don't have enough supplies, they're going to overwhelm you eventually, and they're going to win. And yeah, our system can be overwhelmed, unfortunately. Okay, so mononuclear phagocytes. Oh, crap. Oh, well. The answers are right on the slide. Maybe I left it on mine, but cut it out of yours, right? These guys labeled on your slides? Probably not. So, again, stole from the cute website, right? So this one is awesome, right? Because look at him. Doesn't he look like an eater? He looks like he would bite you, right? And that's what he's going to do, right? Their name, mononuclear phagocytes. So mononuclear referring to their nucleus as being singular and intact. And that's one of the things I didn't mention back here. So this will really help you guys out in anatomy and physiology. All your granulocytes, of course, have granules, but remember neutrophils, their granules don't stain real well. But what's the key giveaway that you're looking at a neutrophil? The multi-lobe nucleus. Do you see this gray, purplish gray, I don't know. I think I'm going colorblind or something. But do you see this nucleus, how it's not a single uniform structure? It's what we call lobed, right? It's been distorted. And this is true for all the phils, all the granulocytes. They all have at least sometimes just stretched out, right? What they call two lobes. A lot of times for basophils, you won't even really see their nucleus because it stains about the same color as the granules, so it's really hard to differentiate. And this is a problem that A&P students will run into too, is that under oil immersion, like 1,000 times magnified, if you look at a lymphocyte, its nucleus itself looks kind of granular, but you really got to look and see if you see the little tiny bit of light blue cytoplasm, right? Then you're looking at a lymphocyte. The chances, too, of seeing a basophil are extremely slim, right? They're not very common in your blood unless you're dealing with something like, allergic reaction. So your slides, it's going to be 1 in 100 in A&P lab that you might actually run across a basophil. And the slides, some of them are so old, you can't really definitively say, right? Of all the years that I taught, I maybe thought I might have saw 1 or 2. You'd see Miss Erica do the happy dance, so very rare, very rare. More than likely, if it's purple, you're looking at a lymphocyte. Look for the light blue cytoplasm. And if you're not sure, ask your teacher or draw what you see and argue it on the practical exam. Right? I always made my students draw. Make sure we were both seeing the same thing. So these guys, right, have got those deformed nucleuses. These guys, as their name implies, mononuclear, right? It stays intact. And phagocytes, these are our eaters. So, the dendritic cell, he's so cute too, right? He's got teeth too. He's an eater. But he's a little different. We'll talk about that. So monocytes, right, are what we call them when they're circulating in our blood. But these cells develop, right? They become more aggressive and powerful and different abilities when they're in our tissues. And the main ability, of course, is destruction, eating it up, phagocytosis. So macrophages are really important. And so we'll spend a little bit more time going into a little bit more detail on them. And dendritic cells are really important for our adaptive immune response. So again, we'll really talk about these cute guys again in much more detail. And what they're really trying to show you here too with this cartoon is that they're named after dendrites because they have these long cytoplasmic projections, right? So they can really stretch themselves out and literally like go grab stuff and bring it towards them. So for your mononuclear phagocytes, especially as it applies to macrophages, they were given names by the location that they reside in our body. So probably the best example are Kupffer cells in our liver. They're actually just macrophages that their home is the liver. So they're not any different from the manganese cells that are in your kidneys. They're macrophages. They're going to eat stuff that comes in, and they're going to clean up debris. Just when scientists were first studying these organs, they saw these cells, and so guess what they did? They named them. In the case of Kupfer, it was named after the dude who discovered them. But come to find out, right, they're all macrophages. They just live in our organs. So thankfully, when we got to naming others, right, we just pretty much started naming them after where they reside. So alveolar macrophages, just to denote that they're ones that live in our lungs. Right? They're not any different from a macrophage in our liver. Just a different home. That's it. So dendritic cells, these guys are super cool. These are sentinel ones. So they don't necessarily have a home like macrophages. Like they'll go visit their cousin in the liver and then travel over. And they're scouting out the scene all the time. They're traveling. But the difference is, what does a scout do? Right? So we're going to send them out in front of the troops, right? What is he looking for? He's looking for the bad guys, right? If he finds them, what does he do? He doesn't run and hide, right? He runs back to his team and tells them where the bad guys are, right? So same thing here. They're going to go out, right? They're going to find the bad guys, and they're going to bring that information back to home base. So in order to do that, they also are going to do just like macrophages. They're going to take the stuff in. They're going to engulf it. But they're the major players that are going to share this with our adaptive immune response, right? They're going to pick up those pieces and give it to the adaptive for inspection. Say, oh, look what I found. Do you recognize this guy? Right? How bad is this one? Have you seen it before? Right? And that's the cool thing about the adaptive immune response, is we remember what we've seen before. That's why vaccination works, because we're going, here, this is what the bad guy looks like. So guess what? When he gets there, your system is ready. You already have antibodies, right? You already have cells that know it's a bad guy, and they're going to attack aggressively and fast. They're not messing around. And again, they usually develop from monocytes, but not always. So then we get to lymphocytes, our last group, which will probably be our last group for today. They're responsible for our adaptive immune response. So we're going to go really big into their specific roles and all the different types when we get to adaptive immunity. But what are the three main cells that are referred to as lymphocytes? You see some of them here, right? The T cells, right? And they can be helpers or they can be killers, right? But who is this guy probably? That's a B cell. And then we have one more not represented by my cartoons. He has the coolest name. The natural killer cells, right? 1973 was a good year. If you watch the other video, right? That's when that cell was named. So B cells and T cells, which are B lymphocytes or T lymphocytes, we just abbreviate their names. For them, they have receptors, so especially the B cell, they're showing it here, right? The B cell receptor is actually an antibody. These guys are not showing their receptors, but they have them. They can recognize specific antigens. We're talking about E. coli 0157, that's specific, right? That specific strain of that specific bacteria. And they generally reside in the lymph nodes, hence their name, lymphocytes, in our lymphatic tissues, and travel through our lymphatic vessels. Not really a superhighway. Instead, the drainage system, right, is our lymphatic system. So then, our natural killers, the difference here is they lack specificity, right? they can't recognize a specific invader. But they obviously are good at killing. So something tells them who to kill, right? But they don't figure that out on their own. So next time, right, we're gonna go add in a little bit more on our neutrophils, our monocytes, talk about cell communication, and I'll probably post another PowerPoint too. The next section we're going to go into is the actual physical barriers. So we're actually going to kind of start at the beginning of the chapter next time. And I'm posting this one for you guys right now.